metabolism. Parang ang careful ko sa boses ko ngayon. Narinig ko yung ni-record ni Zarina kahapon. Sabi ko, ay, ang pangit ng boses ko sa Google Meet. Anyway, so ito yung sinasabi ko sa inyo kahapon na growth curve. Remember that when we're talking of growth with prokaryotes, we're talking about cell increase in cell number. So that's the replication. And sabi nga natin, they reproduce through mitosis so that one daughter cell will be, one parent cell will be producing two daughter cells, di ba? So that in a balanced environment, sabi natin, if the nutrients, what are those requirements? You have the physical and the chemical requirements. Ano na yung physical requirements ng ating microorganism? Ano ulit
Temperature,
yun? Okay.
pH,
Temperature,
osmotic.
pH, and osmotic pressure. How about for the chemical? How about for the chemical? Ano naman ang kailangan natin? You need the
Oxygen,
Oxygen, nitrogen.
and
Carbon.
ang daming problema ng online. Ngayon naman, naglo-lobat ang cellphone ko. Dito ang audio ko. Okay, so you also need your carbon dioxide. Okay? So, ito yung growth curve. So that if everything of these are provided to the microorganism, then the growth of the microorganism then will be assured. Meron siyang, the microorganism will be able to replicate. Okay? Mag-multiply siya. So these are this is the growth pattern or the growth curve. So that you see that there are 4 phases here. You have the log phase or your LAG. Then you have the logarithmic or the exponential, wherein there is an increase already in the number of cells. Then after which, makikita nyo, meron ulit siyang plateau. That is what you call now your period of equilibrium. And then, afterwards, mapafollow na siya ng period of decline or your death phase. So that, here in the phase of your phase 1, your lag phase, which we can also call your phase of rejuvenescence or yung sinatawag din natin phase of physiologic youth. Bakit siya tinawag na phase of physiologic youth? Now, because this is the period wherein nilipat mo si microorganism, you're trying to let the microorganism grow into a different environment. And that environment would be your culture media. And in that culture media, you provide all the needs of the microorganism or your bacteria. So that during that lag phase, nag-a-adapt pa lang yung ating bacteria sa kanyang new environment. So this can be characterized by meron ng konti, little, or no multiplication yet. However, during this phase, the cells in this phase are very active metabolically. So there is already an active synthesis of enzymes and other essential constituents already occur during this phase. So that after nakapag-adjust na si microorganism natin, so it gets now into an exponential increase. So you see the steeping of your curve. Bigla siyang tumaas. That is why it's also called your logarithmic phase or it is your exponential phase. Kasi nga, biglang dumami na sila. So that during this period, the cells are in a state of balanced growth, now characterized by maximum rates of cell division and mass increase. So it is during this phase, during this your exponential phase, that the generation time is constant. Now, after that, nag-multiply na siya, you had that peak there of the peak of the curve, then you get into your stationary phase. Nagkaroon ulit ng plateau. Kaya yung stationary phase mo, this is also your plateau phase, or it could also be your phase of equilibrium. So that during this phase, it is the period at which rate of cell reproduction equals the rate of cell death. Because why do we say this? Growth ceases due to, first is of course, you have, anong una dyan? Sabi ko kahapon, na-mention ko na yan. Bakit tumigil na ang kanilang pag-multiply? O bakit tumigil na ang grow ng microorganism? Ha? Anong unang mangyayari dyan ngayon?
Wala nang nutrients, ma'am.
Wala nang nutrients. Naubos na yung, nagamit na nila lahat. They have multiplied. So that marami na ang gumagamit ng nutrients na nakalagay dun sa ating culture media. So, na-exhaust na lahat ng nutrients. So that in the consumption of nutrients, what happens? Parang tayo, kain ka ng kain. What happens after you eat all of those? May input, may output. So that
May,
is not,
Ma'am,
yes,
may ano naman,
meron na
accumulation
sila.
ng mga toxic products or may mga changes na sa kanyang physiological conditions.
Yes. So that nag-take in siya ng nutrients, meron siyang waste products. So there is an accumulation of these waste products, and these waste products could be producing now yung toxic environment ng organism natin. And of course, because of these toxic products, because of these toxic products, meron ng change ngayon sa kanyang physical environment, specifically with the, physical. Nag-change siya nag-change itong physical requirement, because of the accumulation of these waste products, which could be now toxic to the microorganism. Ano yung physical requirement na yun?
Temperature, ma'am.
Temperature cannot be affected by the waste products. Ano ang maapektuhan ng waste products?
pH.
pH. The pH will be affected because of these waste products. Okay? So that after that, exhausting all of those and changing the pH and everything, papasok na tayo sa death phase or the phase of decline. So that this is now the period at which complete cessation of multiplication occurs such that the death rate in the medium increases rapidly. Okay? So, mawawala na. That is why usually in the laboratory, if you want to maintain growth of your microorganisms, like what we do, is you have to transfer them at least every 2 weeks. Kapag katapos nito, naka-inoculate ka dito ng bacteria, nag-grow siya during the week, hindi mo siya kakailanganin for any lab activity, you have to now transfer that to another medium. Again, nagagawin mo yun for the whole semester, and sometimes we are not unable to do that. Okay? So, yan ang ating growth curve. So that now, we would also want to know, ilan na ba? We're talking about the growth of our microorganisms. So how can we also determine the cell number? Okay, why cell number? Again, let's go back. We're talking of your prokaryotes, and growth would mean an increase in the cell number. So we can do this either by direct counting under the microscope. We can do this by direct plate count. And we can also do that by density measurement. Yung aking cursor. Okay. So when we're doing direct counting, in here, you estimate the number of bacteria. Estimate kasi ang hirap naman magbilang nitong bacteria. Just in contrast to your red blood cells or your white blood cells, wherein you will give them a real number. Sa organism, kasi we're doing the colony forming units pag ganung colony ang binibilang mo. Or later on, we'll discuss on that. So, you estimate the number of bacteria present in a specimen. The problem with this is that it does not distinguish between live and dead cells. Okay? Now, in direct plate count, so we're talking now of direct plate count, you can do dilutions of broth cultures on agar plates so that the unit there, when you're going to count for the colonies, is that you are counting it, the unit would be colony forming units per milliliter. So that is your CFU per ml. Now, this direct plate count will provide a count of viable. Ibig sabihin, we're only talking of the live bacterial cells. Whereas, why viable? Kasi hindi naman na siya tutubo dun sa plate pag siya ay patay na. Gets? Kasi nilipat mo siya you're doing a direct plate count, so you're trying to grow them on a plated media, so that if it is already a dead cell, then that wouldn't grow anymore. So that when you're doing plate count, this one will provide you a count of viable cells only. And it is also determining the bacterial cell count. This is what is used in bacterial cell count in urine cultures. Now, the other method would be density measurement. So when we're talking of density, this is now you had correlated to CFU per ml of the culture. Then this is usually used in the preparation of standard inoculum for antimicrobial susceptibility testing. That is like, for example, the standard that is used for AST is your McFarland standards, wherein your basis there would be to check for the turbidity of your sample before you do your AST. Okay? Get so far? Second slide pa lang naman natin. So, sa direct measurements of microbial growth, do sa plate count, so di ba sabi natin, this measures the number of viable cells, and unit of reporting would be your colony forming units. Now, it can be done in several ways. You can do serial dilution, and then you do either the pour plate or the spread plate. Ano naman si pour plate at saka si spread plate? So, from the word palang pour plate, is that you're pouring the suspension into the Petri dish. Okay? Pinor mo siya doon before you add your cooled media, the temperature before it solidifies. Kasi pag na-solidify na siya, hindi na natin siya mapopour doon sa ating Petri dish. Whereas the spread plate is, andun na yung iyong solidified media, then you put now your bacterial culture, and then you spread it. Usually kasi dito tayo nagkakaiba pag BS Bio or sa MedTech. Spreading in the BS Bio, microbiology, they still use the L-shape na glass rod ba yun? Alam niyo yung glass rod na ganun na pag ininit nyo, mabend nyo siya, magawa nyo siyang L-shape. Yun ang ginagamit na pang spread kasi ipapatong mo lang yun dun sa plate na nilagyan mo na ng specimen, and then you just try to spread the sample. But in our case, usually sa mga medtech, we're using now either your cotton swab or an inoculating loop to spread. Okay? So whichever, basta yan, i-spread mo dapat uniformly yung specimen onto the plated medium. So, yun yung sinasabi natin serial dilution. Why do you need to do serial dilution? You look at the picture here. Dun sa original inoculum mo, so you prepared your bacterial culture. Now, in that original inoculum, syempre hindi natin alam. But you can already, when you get into the laboratory, ang pagtingin nyo pag heavy ang growth is depende sa turbidity. Parang kanina, naggagawa kasi ako ng water because of my sore throat. Nilalagyan ko ng lemon. I did that yesterday. Hindi ko naubos yung tubig ko. Ngayon, turbid na siya. So, ayoko na siyang inumin. So, mamaya tignan nyo, the turbidity, as it gets darker, then the density of your microorganism is greater. Lighter, mas kokonti. Which you can see here now, like in your first dilution, which is 1:10, so that you take note here that you have 9 ml of your broth. That could be your nutrient broth, that could be your trypticase soy broth, the different broth media that are available, or what we are using in the laboratory, so that when we say it's a broth media, then yan yung liquid media. So your tubes here have 9 ml already of broth in it. Okay, so that you start with the original inoculum, that you get 1 ml of that, you transfer it to your first tube, so that your dilution there will be 1:10, and so on and so forth. This is what you call now your tenfold dilution. Kasi if you look at the dilution, your first tube is 1:10, then you have your 1:100, 1:1000, and so on and so forth. So that when you try to inoculate this onto your plated media, on your plates, then you will see now na pag doon sa 1:10, walang separation, you cannot see a separate colony. Meron siyang growth sa lahat, na patong talaga siya. Hanggang, so you get into that dilution where you can see clearly the separation of growth. And each growth there, pag ganyan na sa mga plated, you call each growth a colony. So that here, if you were to count all of these colonies sa 1:10,000 dilution, kita mo silang hiwa-hiwalay na, right? And the calculation of that will be the number of cells you have counted, you multiply it with your dilution, will give you now your colony forming units per ml. Gets? Any question up to this point? So, looking at this, anong ginamit natin na method dito? Was it the pour plate or the spread plate? Para malaman ninyo. Let's get into the next slide. So, this is your pour plate and the spread plate. So, sabi ko kanina nga, you already also have your bacterial culture or your bacterial dilution. Sa pour plate, ilalagyan mo muna siya ng specimen mo bago mo i-pour yung iyong melted nutrient agar. So, this is just a general agar. You'll learn about that later on sa mga types of culture media din. So, bago siya lumamig to the point of, bago siya dumating doon sa point of solidification na niya, so i-pour mo na siya doon na mafe-feel nyo naman yan doon sa inyong flask kung okay na yung kanyang temperature. Kasi, what will happen if we pour it onto the microorganism na mainit pa siya? What will happen? Anyone? Ha? Mamamatay yung microorganism. So, wala tayong mabibilang na microorganism. So that after you have poured your nutrient agar or any culture media that you will be using, isi-swirl mo siya to get a uniform distribution of your sample. So that you will see now, sa pour plate, your microorganisms will be seen on and in. Okay? So that kahit i-flip nyo later on yung Petri dish nyo, may makita pa rin kayo nag-grow. Kasi practically, your specimen is mixed with your media. Kaya siya tinawag na the pour plate method. Whereas the spread plate is, sabi ko kanina nga sa inyo, you already have your solid media on the plate. Now you put your sample, again, that 0.1 ml or either 1 ml, or you can do a one loop full of your microorganism, then you spread that evenly onto the surface. So if you just spread it onto the surface of your media, you would expect that your colonies will grow only on the surface of your culture media. So that you will be also now trying to count lang ilan colony ba ang present dito. Okay? I'm just thinking of how we can do this one para at least ma-idea kayo. But I'm just considering the biosafety points dito kasi baka mamaya nag-grow, hindi ko alam kung paano nyo i-dispose of your microorganisms kasi we can prepare a culture media from your place sa mga bahay ninyo. Anong pwede natin gamitin? Anong pwede natin gamitin sa bahay as a solid culture media? Isip. Pwede yung agar. Thank you, Brandon. But now, aside just from the agar, di ba yung gelatin, yung agar-agar na ginagawa natin, aside from the agar-agar, pwede nating lagyan yan ng beef broth. Remember that you get into the composition ng nutrient agar, meron siyang peptone, meron siyang something. So, na-mention kasi sa isa sa mga webinars na na-attendahan ko, yun nga nilagyan nila ng beef broth. Alam mo yung nor? So I was thinking of us doing that experiment and then we'll try to see. Pero tignan ko muna. I'll do it first before I let you do it. Okay? So, para at least makita nyo yung growth ng mga microorganisms on a solid media. Okay. So that's your pour plate and that's your spread plate method. All of which, this is counting of your microorganisms or the bacteria. So we can have also filtration. What is filtration naman? This is usually done for water analysis. Kasi you would like to know kung ilang coliforms ba ang present dun sa tubig ninyo. So are you drinking potable water? So, kasi pag sinabi natin potable, drinkable yan, wala yung kasamang coliforms. So that ito din, from the word filtration, alam niyo natin yan, di ba, yung magsasala ka. So you're going to use a filter paper, salain mo yung sample mo, then you have you submerge that in sa plate na meron din culture media, ilagay mo yun doon, and then you will just check for yung di ba yung filter paper, pinatong mo dun sa media, yung ipro-provide pa rin ng media yung nutrients na kailangan ni microorganism in order to grow. But you'll be counting them onto that filter paper. Okay? So you also have your MPN or your most probable number method. This is based on the fact that the greater the number of bacteria in a sample, the more dilution is needed to reduce the density to the point at which no bacteria are left to grow in the tubes in a dilution series. Okay, so in a direct microscopic count, this is the measured volume of a bacterial suspension is placed within a defined area on a microscope slide. So I don't know if you have gone already to this, kung meron na kayong, kung saan na kayo sa hematology. But this is practically the same, like counting your RBC or your WBC on your Neubauer counter. So ganun lang siya. It's just that dito sa sa micro, sa counting, you're using your Petrov Hauser cell counter. Ito siya. So parang ganyan siya. So that ganito din kasi yung parang ganito din yung Neubauer ninyo. So that you will also count them dun sa mga grid. An upgrade, di ko alam kung tama yung word na upgrade. Similar to this would be the use of Petri films also for coliform. There are Petri films specifically for each organism. Ganyan ang itsura niya. So para lang siyang papel, Petri film. And then pag doon ka nag-inoculate, so ganyan ang itsura niya na babasahin mo sa microscope. Okay? Sorry. So now, we get into estimating bacterial numbers by indirect methods. So sabi ko nga kanina, when you talk of turbidity, you're talking now of, di mo talaga mabibilang si microorganisms doon. But visual, tawag nyo doon, pag yung sa mata lang, tinitingnan nyo kung gaano siya ka-turbid over, of course, a dark background para ma-measure nyo yung turbidity visually. So that, sabi ko kanina sa inyo that more turbid it is, the greater the number of your microorganism. So you can only check for the density of these microorganisms by using now a spectrophotometer. Okay? I hope you were able to at least have an experience with the spectrophotometer in the college. But if not, we can always have that done later on when the situation will allow us. And also here, we have this metabolic activity. When we talk of metabolic activity, this will be well demonstrated when we will be doing our biochemical test. Because when we're talking of metabolic activity, we're talking now of how the microorganism will be utilizing the different chemicals. Para yun sa mga enterobacteria chain natin, you differentiate them whether they're lactose fermenters or non-lactose fermenters. So whether they're using a separate chemical as a source of nitrogen or as a source of carbon. So that's more of the metabolic activity of our microorganisms. And of course, you can also estimate them by dry weight. If you go back to the previous lecture when we said how much of carbon is present in the dry weight of your microorganism, or how much of nitrogen, and how much of phosphate, parang phosphate lang natandaan ko, and sulfur, which contributes 4% to the dry weight of your bacterium. Okay? So that will end now the growth, bacterial growth at topic. So we'll proceed now to the laboratory activity really that we're supposed to have today. So your activity on the bacterial smear prep, again, I would like to commend everyone for a very good presentation. I came up with some observations, kaya lang nagmi-mix na yung utak ko kasi kagabi, I cannot really divulge to you, pero medyo nagkaroon ng, pero hindi ko, meron mga ibang disturbances. So I wasn't able to really get into all of your bacterial smear prep, but I can really commend everyone for coming up with very good presentations. Now, the first comment there is you could have saved on the presentation kasi you didn't really need to get into the procedure kasi nandun niya sa module niyo. It was more of performing kaagad the experiment. Now, when we are in the laboratory, I would just like to remind you of your GMPs or your good microbiological practices. So first is that before you start any experiment, it's always a must na dapat you always have to disinfect your work area before and after every procedure. Okay? And the disinfection is, of course, the first step when you get into the laboratory. Let's just imagine, di ba, pag pumapasok kayo sa laboratory because you had laboratory classes naman na din sa clean lab. What were the observations? Di ba sabi natin lagi nun na pagpasok nyo kaagad doon, dapat ang inyong mga gamit nandun muna sa lagay ng gamit. So, pumasok parang, imagine nyo na sa lab tayo ngayon, may lab tayo ng micro, so dumadating na kayo. Ang una na pupuntahan nyo would be to put your bags and everything dun sa lagayan ng bags, right? Because dapat walang ibang gamit sa ating mga countertop except those that you are going to use for the activity. So that before you get to your countertops or dun sa mga pwesto ninyo, you're already with your lab gown and whatever PPE that would be needed for the experiment. Now, we don't need to be too OA sa mga PPE because you have to do a risk assessment. Ano yung risk assessment? Ano ba yung gagawin kong activity? What are the risks involved in this activity? So that you would know what PPE you should be wearing. Okay? So, nandun na kayo. So, you disinfect now. Nandun na kayo. You disinfect. Like, for example, you're doing your bacterial smear prep so that all of your gamit, yung mga gagamitin nyo, syempre, ni-request nyo na yan following the protocols of the college. And then, na-request nyo na yan. Nasa countertop nyo na yan. Kung ano yung proseso natin. Imaginin nga natin, nasa lab tayo ngayon. So, nasa table nyo na. Nasa positions nyo na. Na-out nyo na kay Ma'am Ming. So that, before you start with your procedure, one GMP is that before you get to your laboratory class, nabasa nyo na yung procedure para naka-imprint na sa utak nyo yung organization ng gagawin nyo. Di pa kayo mamaya pupunta sa mga kasama nyo. Eh, pahiram nga ng marking pen mo, may slide ka, meron kang tissue, meron kang ganito, which is not very good for someone or for you already kasi meron naman na kayong experience ng mga lab activity. Okay? So that if you have read the procedure and meron kayong gustong i-clarify, you can do that before you start touching anything dun sa mga gamit ninyo sa lab. Okay? So, ano pang hindi po nabanggit? So, you have your PPE, then you have disinfected your working area. Disinfection is, of course, before and after. And I commend those who also have to advocate hand washing before and after. And yung iba pa, I'll just try to send feedbacks din personally to all of the, depende dun sa mamaya ng group presentations ninyo. So, now, in the bacterial smear prep, di ba nakikita niyo dito, I just pick up. So, you have different preparations here. Now, I would like you to comment on the first picture to your left at saka yung first row ba tawag dyan? Manamberan natin from the left. So, 1, 2, 3, then 4, 5, 6. So, I want you to comment on the smearing of 1 and 3. Wag na mag-recite yung mga nag-recite kahapon, ha? Dito yung aking listahan. Sana yung listahan ko. Iba naman, si Brandon, napaka-active niya ngayon dyan. Si Dexter. Comment on slide 1 and 3. Yung mga hindi nag-ano kahapon, hindi nag-recite kahapon. Anyone? Yung anyone, yung mga hindi nag-recite kahapon. Unmute. Parang wala kayo. Hello? Hello? Notebook ako, meron akong logbook na mga ano yun. Queen Millengen, you there?
Ma'am, yung slide lang or yung buong picture?
Hindi, slide lang. Yung number 1 at saka yung number 3. Queen Millengen.
Ma'am, di ba may dapat yung diameter nung nung specimen, ma'am, dapat tulad nung sa number 3. Parang mas maliit yung number 1.
Okay? Or another one. Yun ang observation ni Queen, ha? It is in the size of the bacterial smear. O, iba naman. Kasi dalawa itong tinitignan ko yung mga nag-recite kahapon. Kevin. Wala ba si Kevin kahapon? Kevin. Talon. Meron na si Kevin kahapon. Huwag na si Kevin. Julie May. Corpus.
Hello, ma'am.
Yes, Julie May. Anong observation mo dun sa picture 1 and 3?
Ma'am, sa first picture po, nilagay pa lang po yung yung bacteria po. Tapos sa picture 3 po, naka-circular na po siya, na-spread na po siya. Yun po.
Okay. Last one. Mark Said. Nasaan ka, Mark? Mark?
Hello, ma'am.
Yes, Mark. Good afternoon. Kumusta? O, sige, ikaw, Mark. Ano observation mo?
Ma'am, dun sa first picture po, gaya parang sinabi nila, maliit po, tsaka parang ini-spread pa lang. Tapos dun sa next picture po, parang wala pong bacteria sa gitna, ganun, ma'am.
Mark,
Parang
bakit wala kang pangalan sa listahan ko?
Ala.
Wala. Dito sa aking logbook. Mark. O, sige, Mark. Tapos, so ano
Ma'am,
observation mo dyan?
ma'am, sa next picture po is parang hindi equal yung pag-spread, ganun, ma'am. Kasi sa gitna parang walang laman.
Kasi hindi pa siya tapos. O, sige. Joke. Okay. So, what did you miss in all of this? Nakakonsentrate kayo, get out of the box. We're not talking about the size. Pag nag-prepare kayo, parang mamaya, meron akong in-upload na video sa inyong, sa lens natin. Kasi while I was trying to prepare my presentation for today, nag-surf ako, kaya siguro nagloko kanina, puno na yung aking Now, you get out of the box. Okay? In the preparation, while we are saying that you have to follow yung bilog, kasi sabi nyo, tignan nyo yung nasa gitna ngayon, hindi naman siya bilog, but that is still a bacterial smear, right? And if you compare that with the others, sabi mo na pinakamaganda ngayon, yung nasa gitna, yun nga lang, hindi nga pinalo yung dime na sabi natin, or penny size of your bacterial smear. Okay, now, what you miss there is, kitang-kita ko kaagad kanina. Tingnan nyo yung nasa number 3 ninyo, naka-glove siya. Tingnan nyo si number 1 nyo, nasaan yung PPE niya? And when you're doing your bacterial smear prep, alam nyo na ba kung ano yung ini-smear ninyo? Pwedeng alam nyo na, pwedeng hindi nyo alam. Pwedeng yan ay pathogenic, pwedeng yan unpathogenic. But then, I was telling you earlier about the GMPs, that if you are in the laboratory, you will be treating all specimens, whatever it is, as infectious, so that your PPE would always start now with you having your gloves and your lab gown. So, dyan pa lang sabi ko na ganyan. Yung una, wala siyang gloves. Yung pangatlo, wala siyang gloves. Pero what were your observation? Because you were thinking of the procedure from 1 to 4 na dapat ang size niya ganito. But then you have ignored your biosafety protocols, right? So now, look at this slide in the middle. Now, in the slide naman ngayon is that, if you look at it, hindi naman siya bilog na bilog, pero you look at the distribution. You look at the distribution now, you could say that, kung ako mag-aano yan, I could already say that that's a very good bacterial smear. It's evenly or uniformly distributed. I guess, visually, the thickness is good. And then, the only thing there is, sabi na naman natin, hindi naman siya nag-smear, pinapakita na lang niya siguro yung heat fix slide niya or yung heat fix bacterial smear niya, kaya wala na siyang gloves. Okay? So, dito naman sa in your preparation of bacterial smear, I noticed one of the groups, when you're collecting, kasi wala yata, may nag-mention sa inyo ng aseptic technique, that in microbiology, your aseptic technique is a must, is very important in whatever activity you're doing. And before ko makalimutan, di ko rin alam kung sa yung nilapag ninyo ang inoculating loop ninyo sa table, which shouldn't be. Kasi remember that kahit yan ay na heat na pass mo sa flame, you have the aseptic technique, thereby killing whatever microorganism that was there, hindi pa rin dapat yung mga yan nilalapag sa table ninyo. That is why now usually you have your test tube rack. Diyan natin lahat ipapatayo ang inyong inoculating loop, ang inyong tube, para at least hindi nagko-contact sa inyong table top. So, yung slide dito na merong nagko-collect siya ng sample from a tube, take notice of the position of your tube. Going back now, recalling your sa history natin in the spontaneous generation. So, why is the tube tilted here? Ganyan dapat ang practice natin later sa microbiology. You need to tilt your tube para, bakit nakatilt yung tube? Na instead of tilting the tube noong panahon ni na Francisco Reddy, tsaka panahon ni Nespalanzani, or sino pa yung mga kasama nila, si Pasteur using, si Louis Pasteur ba yung sa Swan Neck?
Yes, ma'am.
Bakit nyo tinitilt si tube dyan ngayon when you will be in the laboratory and be performing experiments, picking your microorganism from a tube media? Bakit kailangan siya naka-tilt? Si Lorraine. Lorraine, where are you? Wala si Lorraine? LM. Kasi ba si LM kahapon? Wala. LM.
Ma'am.
Bakit doon sa picture, bakit naka-tilt ang tube habang nagko-kolekta siya ng ng kanyang sample?
Ma'am, guess lang.
Yeah, go ahead. Walang dito.
Ma'am, kasi po dun po sa history noon is yung swan neck po na experiment, ma'am. Pag naka vertical po is may may possibility po na may pumasok pong ibang microbes dun po sa tube, ma'am. Tapos tinitilt din po yung tube, ma'am, para pag kunwari sa heating po, ganun po, ma'am.
Okay. So another point here is, tama ka, avoid contamination kasi hindi mo alam mamaya kung may pumasok. Kasi syempre, considering considering the law of gravity, mahirap makapasok yung microorganism pag nakaganon. Babagsak man yan, hindi siya papasok directly sa loob. So that's just avoiding contamination. And take note of the Bunsen burner that's beside the usually in microbiology, when you're working with microorganism, the closer you are to the flame, the better. Pag wala na close na mamatay yung microorganism natin. Para at least parang lang ba yun? Pag nag-aseptic technique kayo, hindi malayo kasi yung paggalaw ninyo, pwede rin may matouch kayo or unconsciously makontaminate din yung surrounding ninyo. Okay? So, you had your samples from a tube and also you have to collect from a plate. Now, make an observation dun sa plate. Yung nagpipick siya ng colony sa plate. Si Mikey Madela. Mikey. Wala si Mikey. Pangit ang signal mo, Mikey. Si Nolan ba nag-restart kahapon? Nolan, wala ka kahapong receipt. Ay, meron. Huwag na si Nolan.
Hello, ma'am? Ma'am, ano po?
Hindi, may recitation ka na kahapon.
Ay,
Iba
sige,
naman.
ma'am.
Robert. Si Robert. Nandyan? Robert? Shane Ocampo.
Ma'am,
Yeah, Shane. Make an observation dyan sa ating panglima, yung sa plate. Yung nagpipick up siya ng colony sa plate. Observation.
Ma'am, naka-slant siya.
Ang alin?
Yung agar plate po.
Okay, naka-slant siya. Bakit siya naka-slant? Di ba pag ganyan, tignan mo siya. Anong sabi natin kanina sa tube? You keep it, naka-tilt siya para hindi pumasok ang microorganism. Tignan mo si plate niya. Or was this only for demo purposes?
Ma'am, para din po, maka-prevent siya ng drying out saka contaminants.
Take note ha, tignan mo siya, nakababa yung kanyang plate cover, naka-open fully ang kanyang plate.
Opo, ma'am.
Tama siya o mali siya?
Ma'am, pag fully open po kasi yung agar plate mo, siya. Malaki yung possibility para maka-enter yung mga airborne contaminants.
Tama ka dyan. Open na open siya so that any microorganism can get into the plate. So, although this could just be, ito siguro is only for demo purposes of how to pick up. Pero meron kasing technique ng pag-open ng inyong plate when you're trying to pick up your colonies. Okay? So, if we just have to look into this one, mali yung kanyang pag-pick ng colony. Inopen niya, not unless, excuse me. Ito ay sa loob ng biosafety cabinet. Alam niyo kung bakit nasabi kong nasa loob siya ng biosafety cabinet? You see the grills, itong butas-butas sa harap niya at saka sa likod.
Opo, ma'am.
Sa biosafety cabinet yan. So that, alam niyo yung biosafety cabinet natin, that when you get into the clinical lab, yung nasa kaliwa ninyo, nasa tabi ng inyong lagayan ng mga bags. Nakita niyo yon?
Yes po.
Yes, ma'am.
Do you remember? So that is a biosafety cabinet. So that when you are doing your work inside the biosafety cabinet, like this fifth picture here, yung nasa gitna, you can open fully kasi that biosafety cabinet is a sterile place so that everything that rotates inside, wala kasi tayong topic niyan, pero I'll try to, when we get into that, I'll try to tell you the principle of the biosafety cabinet. May filter yan, yung mga grid na yan. Diyan dumadaan, nagro-rotate, and then it gets to a HEPA filter that's in the biosafety cabinet. So that you can open, you can do this, lahat yan sterile. Wala kang, there will be a very, very, very slim chance of contaminating your plate. But if you were in your countertop, nandun lang kayo sa table ninyo, binigay ko yung plates ninyo, binigay ko yung tubes ninyo, at hindi naman kayo pwede lahat mag-work sa biosafety cabinet. Because that biosafety cabinet, isa lang ang pwedeng mag-work, at the most is 2. So, opening it like this would be a no-no. Hindi pwede yan. Okay? So, I will try to demonstrate later on, mag-video tayo, and we'll see how you should, if you could get, yung makalabas sa inyo later on, I will tell you that you could get disposable plates pag na-risk assessment po na yung paggawa ng media sa mga bahay-bahay natin. Okay? So, this one is picking up now your sample from a plated media. So that when you pick up your sample from a plated media, dapat yung parang tinatch mo lang yung colony. And when we're talking of colony, makita mo talaga yung nag-iisang bilog lang na nag-grow dyan sa media. Hindi pwede yung ganyan na ang dami, hindi mo siya ma-differentiate because you don't know. This would give you a mixed bacterial smear. Okay? So, yung sources nyo were either you can get it from broth or you can get it from a solid media wherein you pick your colonies. Okay, so alam nyo na yan dun sa mga presentations ninyo. But then, now you just have to assess, was your bacterial smear too thick? Was it just right? Or was it too thin? Ano ang magiging effect nito sa inyong gram staining later on pag ganito ang inyong bacterial smear? Remember that the first step in your methods of identification is you prepare your bacterial smear for the appropriate staining procedure. So, paano yan pag tutik yung smear mo? Paano mo malalaman? O paano mo makikita? So that, merong test dyan, di ba? How do you do that? Ano yung test na mention yata yan sa ating sa inyong module. Paano mo malalaman kung just right yung iyong bacterial smear? Tapos na. Sino hindi pa nakasagot? Asan si Unica? Meron ba si Unica kahapon yung recitation? Wala. Unica, where are you? Unica? Wala. C. Bariwan. Bariwan? Okay. Shem, Matalang. Shem? Wala din. Ah?
Ma'am, ano po yan?
Bakit nasan ka, Shem? Si Shem ito?
Opo, ma'am.
Yes, Shem.
Si May po.
Ah?
Ma'am, ano
Shemay.
po yung tanong?
Shemay pala. Bakit di mo
Yes,
narinig?
ma'am.
Shemay? Katulog ka yata, Shemay, kasi anong oras na ba? Paano mo malalaman kung ang iyong bacterial smear ay too thick ba siya, just right ba siya, or too thin? After na-prepare mo na siya, ginagawa ka na, nag-swirl ka na, nag-emulsify ka na, depende kung anong ginamit mong sample. Paano mo malalaman ngayon kung too thick, just right, or too thin? Titignan mo lang siya.
Ma'am, guess ko lang po is, pagkatapos po nun is, parang yung sa detection po ng bacteria. Kasi po pag tutik po mas mahirap po ma-detect po yung bacteria compared po dun sa tin po mas mabilis po yung detection ng bacteria.
Paano mo madedetect? Hindi, wag mo na yung bacteria kasi hindi pa natin na-stain yan. Hindi. Si smear pa lang, preparation pa lang ng bacterial smear. So natapos nyo na yung preparation ninyo. Paano mo ngayon ma-check kung ay ang kapal naman ng aking bacterial smear? Ay, naku, ang nipis-nipis nito. Or, ito lang tama. Before you proceed with your staining procedure. Paano?
Ma'am, po,
Visual?
it allows newsprint po to be read.
Yes. So, ipapatong mo siya sa any newsprint. Papakita ko yan mamaya. Meron ako sa AFB. So, ipapatong nyo sa newsprint. Dapat, hindi naman yung kitang-kita mo yung newsprint. Kasi pag kitang-kita mo yung newsprint, yun yung Shem. Pag kitang-kita mo, pag pinatong mo siya sa any newsprint at kitang-kita mo yung newsprint, ano yun? Siya ba ay okay na yun or hindi?
Pag po, tutin po, ganun po.
Pag tutin. Okay. So, yun yung ninyo. So that you try to put it. Pero pag lagi ka nang gumagawa ng bacterial smear, tinignan mo pala yung pagka-fix mo, sabi mo, okay na to. Okay? So, this would be now, after you have prepared your bacterial smear, dito nyo na makikita microscopically whether yung bacterial smear mo ba was too thick, it was right, or it was too thin. Okay? So, ideally, you should come up with this one. Andyan yung gram positive mo, andyan yung gram negative mo, and also with the ones on your right. Ano ang qualifications nitong mga smear na ito ngayon na nag-gram stain mo na that would tell you that your bacterial smear prep was done properly? Ano yung criteria ba tawag doon niya? Criteria. Ano yung criteria mo ngayon na, okay, my bacterial smear was done properly? Looking at the pictures that you have there now. Parang natawag ko na kayong lahat. O, sinong gusto mag-recite? Julie May? Wala yan sa akin, Julie May Corpus kahapon pa. Julie May. Julie.
Yes, ma'am. Ma'am, ano po yung question?
Asan kayo? Bakit biglang pag tinawag kayo? Ma'am, ano po yung question? Julie May, Sabi ko kanina, di ba, na nag-present tayo ng too thick, too thin, just right. Now, with these images now, how would you say na yung ginawa mong bacterial smear was done properly and you came up with an ideal or a very good smear or a just right smear?
Ma'am, maganda po yung distribution po nung bacteria sa slide po. Parang
Okay.
even distribution po.
It's not just even distribution. You are able to identify the morphology of your microorganism. Walang overlapping, magkakahiwalay sila, mapapakita talaga yung arrangement nila, makikita mo talaga whether it is a cocci, it is a bacilli, or whatever other forms there might be dun sa slides mo. So, kaya tignan mo itong dalawang picture nyo dito. You have now the one on your left, itong purple, wherein meron ba kayong makita na definite shape or size ng microorganism? Parang stain lang yata ito, baka dumi lang ito. Okay? So, in that, you can already see that this one was prepared, it's too thick, and then, tingnan mo naman yung too thin. Di mo alam ngayon kung ito ba ay debris lang ng stain mo kaya nagkaganyan. Kasi wala naman ako makitang koksay o meron siguro doon. Di ko alam kung koksay yun o bacilla yun. Ito ba ay mga leftover, mga debris lang ng stain or wala ba ako talagang nakolekta? And this usually happens pag yung inyong specimen ay galing sa tube. Usually konti. Well, it is better always to get specimen sa colony picking, although kuminsan naman masyado siyang thick. So, it's just in the manner of preparation of your bacterial smear for you to be able to come up with images later on. Pag na-stain nyo na siya, masaserve niya yung purpose niya into presumptively. Remember, presumptively, because your gram stain will not definitely give you now an identification. Sasabihin lang niya sa iyo that it's a gram-positive bacteria or it's a gram-negative bacteria. Or is it, bibigay din niya sa iyo kung yan ay cocci or yan ang bacilli para meron ka na ngayon ma-entertain na microorganism to really test to come up with your definite diagnosis. Okay? So nakita niyo na dyan ngayon kung ano ang importance ng inyong bacterial smear preparation. Okay? So, ay, bongga, meron siyang ganun-ganun. So, that is now with your gram stain. Now, I posted in our lens kanina yung isang activity that I went over na maganda yung presentation niya. And that is one of your assignments. Please, ano lang, that deadlines, as I have said earlier naman, that deadlines are placed there only to push you. But there can always be considerations. Pero wag naman na porket may considerations, ipapahuli-huli nyo na siya. Okay lang naman kay ma'am, later na lang yan. Do your time management properly. And like I have said earlier during our orientation, yung first meeting natin, I am old enough to accept criticisms or whatever, but I would appreciate suggestions from you. Kasi nga sabi ko, it's not really what we present, but how you are able to gain knowledge from what is being given to you. And you do not stop there. Di ba sabi ko, wag kayong complacent ba tawag doon. Yung parang kung ano lang yung binigay sa inyo, yun na lang. So, I really advise you to get into reading more books. I know that toxic nga itong online, but meron naman tayong kasabihan na sa Pilipino na pag gugustuhin may paraan. Pag ayaw, wala talaga. Okay? So it is the motivation of learning more that will push you into trying to read more. Okay. So what is DSSM now? Bakit kailangan ko i-present ang DSSM sa inyo? Ito ang application ng inyong acid fast staining. Diba sabi natin, where do you apply your AFB staining? Is for your Mycobacterium tuberculosis. So, and that is what we call our DSSM microscopy or your direct sputum smear microscopy. Now, ito yung flow lang ng DSSM. So, you see here again, papasok na naman yung preparation ninyo ng bacterial smear. The only difference here is your biglang na pa break your sputum daw. Your specimen will be sputum. Okay? Yung sa ating protocol ngayon with DSSM is that dati kasi 3 days yan. Pero yung bagong NTP in the National Tuberculosis Program is that pwede na ang 1 day na 2 specimens lang. So you will be asked to give yung specimen nila ng upon arrival or pagdating mo, dala-dala mo na from home, then after 1 hour, mag-collect ulit. So ito na naman yung smearing procedure ninyo is the spreading of the sputum, which is much, much different than just ordinary doing your bacterial smear prep. Then it has also the drying and the fixation. So that this has been discussed in the module already. Then you go again to your staining protocol that will have initial staining, heating if it is. Anong nangangailangan ng heating doon sa dalawang method natin ng acid-fast staining? Anyone? Anong nangangailangan ng heating doon sa dalawang method? The most commonly used method in acid-fast staining. Tam!
Ma'am,
Acid Go. Sino yun?
ma'am,
Yeah.
ma'am, Israel, ma'am.
Okay, Israel.
Ma'am Zin Nielsen, ma'am. Ma'am Zin Nielsen.
Okay. Ano yung other method, Tam?
Ma'am?
Ano yung other method?
Ma'am, Kinyon.
Yun, ma'am.
Kinyon. Okay. So, yung pareho for your Mycobacterium tuberculosis. So, ayan yung staining nyo, decolorization, washing, counter-staining, washing, and then drying. And then, of course, you get to your microscopic observation, then reporting and recording, which we will have that later. Kasi nandito pa lang tayo sa staining procedures. Okay? So, ay maarte yung PowerPoint. Meron siyang animation. So, the labeling of slide is very important. And also, I forgot to mention, the choosing of the slide is very important when you're preparing your bacterial smear. Had you been in the laboratory, I would tell you just to get with the frosted slides. Alam niyo yung frosted slides? Pero if you get the frosted slides, may peke din kasi na frosted slide. Pag finil niyo yung frosted end niya, dapat ma-fill niyo yung rough area. Kasi yung mga iba, sabi nila frosted slide, pero pag i-fill mo yung rough area niya, parang wala namang rough area. Smooth pa rin siya. Ano ang purpose nitong frosted slide? Kasi, di ba sabi nyo doon sa mga presentation ninyo, you label with your marking pen. What's the problem with the marking pen? Pag nag-decolorize kayo at napunta yung decolorizer doon, pwedeng mabura si marking pen. Remember that's either you're using acetone or alcohol. Whereas, pag dito sa mga frosted slide, aside sa hindi siya madulas, parang wala siyang grease, pag ginanon nyo ng inyong thumb, finil nyo yung surface niya, yan, ma-feel nyo na hindi siya talaga yung madulas. So that's the best slide to do your bacterial smear preparations. Okay? Aside from that, madaling mag-label sa dulo niya and you can use pencil. Yang mga yan hindi nabubura. It will stay on that frosted part of your slide. So that labeling, you know that already, labeling can be dependent on the procedure. Siyempre, pag simple ang bacterial smear and you were given a known specimen, kung yan is staph aureus, then you will label that, of course, with your staph aureus and whatever labeling that would have been stated in the procedure. And then, of course, sa smearing, ayun yung spreading nyo, sabi ko kanina, iba ang spreading sa AFB staining. Hindi lang yan, basta binilog-bilog nyo dyan, yun na yun. Hindi yun. So that, we're using coconut midrib. Alam niyo na si coconut midrib, yung ginagamit sa walis tingting. Although, you can use already an applicator stick kasi hindi ito madidistribute ng ating, pwede rin siguro ang inoculating loop. But when we're doing AFB, the best there would be your coconut midrib or your applicator stick na pukpukin nyo yung dulo at magmumukha siyang soft broom, walis. Yung ganun. Parang may ano siya. Try nyo mamaya magpukpok ng ng posporo. Kung may posporo pa kayo sa mga bahay-bahay. Okay? So that when you get that, you're going to spread it onto approximately 2 by 3 centimeter. Now, with your AFB, with your acid fast, hindi mo sundin si 2 by 3 centimeter because dun sa reading mo later on, it is now, it gives you the number of field of views that you are viewing when you finish going over the whole 2 by 3 cm na sputum smear. Okay? So, doon sa inyong bacterial smear, pwede kahit ano, kasi you're just trying to represent, but when you're doing your AFB with your Mycobacterium tuberculosis, the size is very important. So, the 2 by 3 size is what you're going to do. Okay, so, after you have smeared it, so we say now, what is a good smear? Ito na nga yung sinasabi ko earlier, that it should be, hindi yan, dami naman animation nito. So this is your proper spreading. And this one takes a skill takes time for the skill to be developed. You're doing coiling, repeated coiling. Hindi mo lang siya, yung ginawa nyo doon sa bacterial smear, di ba? Sabi natin, ano na yun? Emulsify mo lang o spread mo lang, ganun. Dito, isa-isa mong i-coil siya, repeated coiling, yung buong 2 by 3 area mo. So, I would like to ask you later on, iniisip ko, gagawin ko muna because every activity that I would ask you to do, I'll have to try it muna kung effective ba siya just to show itong repeated coiling sa AFB stain nyo. So, ito yung proper spreading ninyo. So, nakikita nyo yung direction ng parang repeated coiling niyan. Okay? So that, yan na yun siya. Allow the smear to dry. Do not dry it under the sun or over the flame. Ito na talaga yung hindi over the flame. Why? Your MTB, because AFB is for MTB, is aerosol forming. That even you're trying to open the container of the sputum, dapat very careful kayo. Hindi nyo yan isi-shake because bursting the bubble, sabi lagi, do not burst the bubble because you're releasing the TB bacilli in the air. So that ito kung hindi completely dry at yan ay dinry mo over the flame, yan ay pwede mag-produce ng aerosol. You are releasing the TB bacilli sa air. Okay? And in here, since you're working with sputum na pwedeng meron o wala, but since this is microbiology wherein we said all specimens are considered infectious, hindi mo basta-basta itatapon ngayon si iyong midrib because you have to disinfect that. So, ididiretso mo yan sa bottle with alcohol, pwedeng sand mixture, or pwedeng Lysol. So, you could use any disinfectant for your midribs. Okay? Okay? Usually kasi ngayon, minsan naman binaburn nila kaagad. But then, that burning could also, syempre basa yung iyong stick, pwede ka rin mag-form ng aerosol. So, the best way would be just to put it dun sa sand mixture mo or sa bottle na meron siyang laman na usually ang ginagamit ko Zonrox. Kasi sa sobrang kaparanoid ko sa micro, hindi ko siya dinidilute. Pure Zonrox kasi mahal ang Lysol. Okay. So, fixation now is that you're fixing the smear over flame. And then, of course, it's the back of the smear and never scorch the smear. Nandun na yan sa procedure ninyo. So, ito yung talagang natin is the staining. Ito yung sinasabi ko sa inyo dati yung So, you arrange the slide consecutively. Siyempre, depende yan. Depende sa method mo or what you have developed in the laboratory. And then, you do your staining. Ang pinaka-importante dyan na sinasabi ko nun sa inyo is itong heating of the slide. Okay? So, yan yung heating of the slide. How do we do this? We do not, di ba, flinad mo na siya with your primary stain. So, you cannot pass that over a Bunsen burner kasi andun yung liquid. So, that you put a cotton into an alcohol, or you hold it with that, with forceps or your test tube holder. Pero tignan nyo muna kung walang balot na plastic yung test tube holder. So, you forceps and then, magpa-flame yung cotton, you pass it over the slides hanggang mag-produce lang siya ng steam. not boiling. Okay? So pag medyo umusok na siya, tanggalin nyo na yung inyong flame under the slides. So yun yung pinaka-importance sa ating AFB staining. And then, of course, the colorization and all is the same with your gram stain, except that you're using your 3% acid alcohol, and that, of course, is made up of your hydrochloric acid and your ethanol. Washing of the slide, that is, of course, sabi ko nga, gentle stream of running water. Wag magmadali at baka mamaya ma-distort pa or pwede rin siguro siyang ma-wash out. And, of course, your counter staining. So, in your counter staining, sabi natin, is dependent on the method that you're using, whether Ziehl-Neelsen ba yan or Quinones. Okay? So, tilt the slide, washing and drying, fast forward na natin. Ito na ngayon yung inyong finished product. Okay? So that after that, you proceed your microscopic examination, and ito na ngayon yung gagamitan nyo ng oil. But since we're only into smearing muna, overview lang ito ng ng pag-read later on. We'll talk more of this later sa Mycobacterium tuberculosis. Since lesson lang natin is until AFB. Yan ang itsura niya, but this is only for demonstration. Hindi siya ganyan kaganda pag tinignan natin sa slide. And ang hirap niya hanapin. Okay? So those are just pictures of an AFB stain that has been properly stained. But I will give you some here of improperly stained smears. So this is one that is properly stained. Then you take note of that. There is insufficient heating. Why are we saying there is insufficient heating? And why do we need heating when we're using the Ziehl-Neelsen? Anyone? Anyone? Bakit kailangan ng heating? Anong purpose ng heating? Hanap na naman ako. Anong purpose ng heating? Wala na kayo? Chi! Kita ko yung pangalan ni Chi. Chi! Joshua, Chi.
Hello, ma'am.
Anong purpose ng heating?
Ma'am, maghula lang po. Magbe-base po ako dun sa...
Ayaw ko ng hula. Joke lang. Sige, go.
Ma'am, di ba nga sabi niyo po, ma'am, i-heat po siya hanggang may makita pong steam, pero not necessarily po na para mag-boil.
Mm-hmm.
Siguro, ma'am, yun po para mawala po yung mga excess na fluid na nilagay po.
Hindi. You're not removing excess fluid.
Okay, ma'am.
Okay. Jericho. Ral.
Ma'am.
Jericho.
Ma'am, ayaw mo ako, ma'am.
Ayan na, nakita ko na nga. Ralph, ano?
Ma'am, ako, ma'am. Harley to.
Ay, si Harley ba ito? Harley, kakanta ka na?
Ma'am, hindi. Ma'am, para
Sige.
po yung yung mga waxy material sa cell wall, ma'am.
Ano meron nyo sa waxy material niya?
Ma'am, kasi resistant siya ma'am, yung waxy material. Resistant siya sa staining. So, hindi kakapit yung stain. So, para kumapit yung stain, kailangan i-heat para matanggal yung waxy material.
Okay. Ano ko lang yung word na matanggal, ha? You're not actually removing. Hindi, tama yan. Okay lang yan. That's how, kasi syempre yun nasa isip nyo. Sabi ko nga.
Soften din, ma'am. Soften niya yung waxy material.
Isoften niya. Okay? Kasi parang isipin mo yung kandila. Pag inano mo yan, pag minelt mo, magme-melt siya, then pwedeng pumasok ang something into that. Para matrap siya mamaya pag nag-ano ulit siya, di ba? Parang ganun lang. So that you're not actually removing, you're just trying or helping. Kasi nga may folic acid yan, fatty yan. Ang hirap, sabi nyo, na pasukan ng stain. Kaya nga, you try to heat it so that you try to heat and then magme-melt yung magsusoften or whatever yung fatty layer na yun para matulungan pumasok si stain. Okay? So, yun ang purpose niya. That is, another one would be, ano yung na-mention natin sa ating earlier lecture, aside from heating, yung wetting agent ninyo, ano yun?
Surgical, ma'am.
Surgical. Very good. Okay. So, ito yung mga ano lang. So, here you also have an under-decolorized, and then this is your intensely counter stain. So, I cannot really comment kasi doing an AFB, if you did not develop the skill, it will start from the preparation of your smear. And usually kasi, pag nagpre-prepare tayo ng smear, medyo, alam mo yung, ano natin, ito yung mga ano lang reporting. Yung pag inopen mo pa lang yung sample, hindi ko alam kung meron ako dito sa ano. I'll skip this one ha, kasi there is a new reporting for TB according to NTP. Kasi nababago ito almost every year, depende pag nagmi-meeting ang ating National Tuberculosis Program Committee. And lagi itong na-update basing din sa WHO. So I'll skip this. We'll just go to that on the, I will skip this too. Ito, pwede ito. So, ito yung mga main causes din ng false reading kasi this will be dependent on your smear. So, that tignan nyo yung size, too big or too small. Sabi ko, you cannot get out of the 2x3 dimension of your sputum smear. The evenness, the thickness, the cleanness is very important. Kasi kung minsan, pag may scratch yung slide mo, tapos nag-deposit doon ang iyong carbofluoxetine, pag hindi ka rin talaga experience, sasabihin mo, ay, positive siya. Eh, yung pala, pag minove mo yung iyong fine adjustment, mawawala siya. Yung pala, scratch lang yun. Okay? And then, specimen quality is also important. Sa NTP procedure ngayon, dati kasi pag salivary, pag nakita mong saliva lang yung specimen, rejected na yan. Ngayon, hindi na. Kasi remember that aside from doing now AFB staining, meron na tayong gene expert din. So, as of the latest, meron lang ibang procedure na dagdag pag saliva ang specimen. And then, of course, you have here yung mga causes din sa staining. You have your overheating, insufficient heating, over-decolorization, or under-decolorization. Okay, skip natin yan. Ito yung smear assessment natin. So, ito yung sinasabi ko na factor usually sa paggawa ng ating sputum smear. Kasi nga sa AFB ito. Siyempre, hindi mo na matanggalin parang, ay kadiri naman ito, lalo na pag bloody yung specimen. Or lalo na pag mucopurulent yung specimen. But since you are health workers, yung mga diri-diri na yan, hindi pwede pag nasa lab na tayo. What is important is, dapat lang talaga na magawa natin yung tamang sputum smear natin for your AFB stain. Okay? So, here, the presence of leukocytes can also be an indicator whether your specimen is right or whether, so specimen ito and sa smear prep natin. So, ito na lang ulit yung mga examples. So, I have slides here pero hindi ko din naman pwede ipakita sa inyo. But later on, you will have to practice on this too. Ito yung too thick. Ito yung sa baba mo yung smear mo. Yan yung sa microscope niya. Pag too thin, yan naman. And then, ito yung sinasabi ko earlier sa inyo na technique on how to check whether your smear is too thick or is it too thin or you did prepare the right smear. Okay?
Ma'am,
Yeah.
may tanong raw po si Ralph, ma'am.
Yes, Ralph. Ralph, go ahead. Post mo sa chat. Ralph, pwede po bang mag-perform ang AFA kahit fresh grad po? Ralph, anong ibig mo sabihin sa magpa-perform? Kasi ito, usually, mahirap, hindi ka, pag wala kang training, hindi pwede. Kasi sabi ko nga sa inyo kanina, from the preparation of the smear pa lang, mahirap na. Okay? And then, there's the issue also on biosafety. Remember, you're working on a sample. And when sa setting kasi natin usually, when you're doing your bacterial smear, your sputum smear, kasi focus tayo sa AFB. Your sputum smear, pag pumunta kayo sa City Health, mananotice nyo na sa labas siya gagawin yung smear. Why sa labas? You need an open air. Kasi nga, for the main reason that your AFB bacilli, which is your TB bacilli, can stay in the air for 72 hours. Kung meron ha, kung meron. Now, doing it outside, pag wala kang biosafety cabinet, again, which will protect you from either infecting yourself, infecting your environment, hindi pwede na maggawa ka ng ganito, ng Mycobacterium smear, if you did not have the proper training. Okay? I had the opportunity to be trained din kasi meron kasama kami sa TB something ng DOH. Ang hirap mag-prepare ng smear. Ang hirap din maghanap ng TB bacilli. Pag i-microscope muna siya. And you will really need the skill to do all of this. So while we give you this experiment, pinapagawa ko ito sa estudyante, pero I do not let you prepare the smear. Meron lang tayong saliva. You collect a saliva just to be able to practice the repeated coiling. Kasi hindi na pwedeng hindi nyo alam yan. Kasi pagpunta nyo ng CVMC sa pagdating ng internship ninyo, gagawa kayo yan pag dumating yung specimen ninyo na sputum. Sabihin nyo, paano gumawa nito? Akala mo bacterial smear lang. It's not a bacterial smear. It's not a, tawag nyo dyan, yung simpleng bacterial smear lang. Kasi sabi ko nga, iba ang smearing niya. I'll try to do a video ng mag-coiling ng sputum smear. Okay? Para at least makita nyo. But I'll take note of that na pagkatapos itong sitwasyon natin na to, we can always try to do all of those, yung mga importante, yung mga gagawin nyo pag nag-intern kayo. Because, of course, the online or whatever, kahit simulated activity or kahit ma-view pa kayo ng lahat yan, if you don't really do it, hindi mo malalaman na ganyan pala yan. So, sabi ko nga kanina, I'll try doing it using either condensed milk para tignan natin pag nag-dry kung makita ba yung coiling or what. Or anything, or pwedeng sputum talaga. Okay? But to that question, Ralph, hindi pwede pag wala kang training. Okay? So, ito naman yung smear cleanness ninyo. Kasi pag maganda yung slide mo, at not only with the slide, even with your staining solutions, when we say that they should be freshly prepared, dapat yan ay na-filter yan para walang mga debris, ay kailangan talagang, it will also affect the cleanness of your smear. Okay? So, in summary, for your AFB, is that a good smear is made from mucopurulent sputum. Not really the bloody sputum. Di ba? Usually, meron tayo yung pag nag-cough out ka ng sputum na may blood. Alam, may TB yata ako. Mga ganun. But the best standard is that a mucopurulent sputum. Kasi i-describe nyo din yan mga sputum sample ninyo. Pag naggawa tayo ng ganito. Okay? Spread evenly and repeated in coil type form. And of course, again, the size is there. It should be 2x3, thin enough to read newsprint through after drying, and it should be air dried before fixed by flaming. Okay? So that's the end of slide. I did not put up examples of special stains because practically it is dependent on the structure that your organisms have. So that hindi rin naman natin ginagawa sa laboratory yan. But it's not because hindi natin ginagawa sa laboratory, is that you do not get to know what it is na. Kasi later on, hindi natin alam. Pag pumunta kayo sa veterans, meron doon si Sir Gami na bigla lang kayong papipilahin lahat. From UST, CEU, FEU, lahat ng schools, CSU, PLT, lahat ng schools, tatanungin kayo. And if you ask your mga ates and kuyas, we have a very good reputation sa veterans and in all the other hospitals. Ah, inaabangan nga lagi yan pag tatawag na yung mga chief tech kung meron akong ipapadala. Kung meron ipapadala ang college na intern sa kanila. Kasi inaabangan nila, magagaling na daw, masisipag pa. O, di ba? Yan kayo. So that you do not just limit yourself. Again, let me reiterate, do not limit yourself to the modules that are given to you. Maraming reading materials dyan. While I understand truly and honestly, kaya nga natanong ko kayo nun, di ba kayo overloaded ng activities? Kasi alam ko this one is the online activities are given there parang to enhance din yung learning experience